Okay, I think we stopped here. Okay. So last week we talked about all the different stages of memory. We talked about a few clinical cases of amnesia and kind of like what that looks like clinically. So today we'll focus more on like the molecular, like how does actually the brain represent this information? When I say that a memory is stored, what exactly does that mean? Okay, which will take the form of molecular, synaptic, and cellular changes. Okay. It might involve changes of the synaptic level, so where the weights of the synapses between two different neurons have been changed, and we'll talk about what that exactly looks like. Or you might actually have the birth of new neurons, okay? And we'll also give examples of that. And all of these physiological changes are examples of the brain's ability to be plastic, or neuroplasticity. Okay? Okay, which is the ability of the brain to change, or the nervous system to change in response to some sort of experience or some change in the environment. And here we're talking about learning and memory. Okay, so what exactly do we mean by physiological synaptic change? Well, you can have increased neurotransmitter release from the presynaptic neuron, or you can have an activation of the transmitter release. You can have a greater effect due to changes of receptors, so you can actually insert more receptors to make a synapse more effective, because the number of receptors are not fixed. We talked about that before. Or you can influence other neurons. And long-term memories, okay, and we talked about what long-term memories, what that means last time, are going to bring on actual structural changes in the synapse by adding more receptors or making receptors more effective, but there can actually be an actual structural change where new synapses are formed or more receptors are inserted. So here is a schematic representation of what that synaptic change is, what that might look like. So on the left side is before training or before learning. On the right side is what might happen at the synaptic level after learning. So in A, you have after training, you might have an increase in neurotransmitter release. So do you see the red dots that represent molecules of neurotransmitter? You see there's more red dots there. Or you can have the postsynaptic side become larger and more efficient. You see that in the middle one? Or you can have both the presynaptic and the postsynaptic becoming larger, and thus more effective. In B, I'm showing here another interneuron modulating the presynaptic side so it can release more neurotransmitter, more red dots. Or you can actually have new synapses being formed. So you see in C, you see how there's a new collateral here? This wasn't there before. This is an actual new synapse that was born as a result of learning or training. Or you can have ones that are not used, like in D. Look at this first collateral, because it's not used. There's no neurotransmitter being released. There's no information being exchanged here. So what we do is we get rid of this collateral that's not being used, and this one grows another collateral because it's being used. Yes?
For part A, is it possible for just the presynaptic terminal to get bigger?
Yes.
Okay.
Yes. Yes. Okay. So these are just some examples of actual structural change. So when I say store information, when I say a memory is formed, when I talk about memory trace, I'm talking about actual changes that happen structurally in your brain that might take the form of one of these. Does that make sense? Okay. So in animals, we can actually test this memory, and we can look at the biochemical changes much more invasively than in humans, where you can have, and we've done this, for example, in rats, the one experiment that's basically in every psych book, where you have rodents living in a standard condition, in a standard cage, and then another group of rodents who are living in an impoverished cage, so no California cheese, no mate to play with, no wheels to exercise on, and then another group that's living in an enriched environment where they have cheese, they have wheels to play on, they have other mates to play with, so something like this. And what we have shown, not just in rodents, but in other animals as well, is that animals that are housed in enriched environments tend to have heavier, thicker cortex, meaning they have more dense dendrites, and they have enhanced cholinergic activity. So if you remember, acetylcholine is important for arousal, for concentration, for focusing. They also grow more dendritic branches and spines. And if you remember from previous chapters, having more dense dendrites does what? How is that important to have more spines or more dendrites? Yes, Sasha?
Information.
Exactly. You can receive more information, have more complex messages. The synapses, the cortical synapses, become larger and more effective, which is consistent with the storage of long-term memory. They have more neurons in areas that are important for learning and memory, such as the hippocampus. And if they were to get brain damage, they recover much better in comparison to the control or the impoverished condition. And basically, we've seen this phenomena from flies all the way to humans. That when an organism, be it flies, rodents, humans, when they live in an enriched environment, you get all these structural changes. Okay? So if, here we're counting the number or the layers of dendrites. Obviously, the more layers of dendrites, the better. And if we compare these three groups on the order of branch, meaning like the layer of branch, what you see here is that for the enriched condition in the fluorescent green, that for layers, starting at layers 3, 4, and 5, that they are much more, they have more branches in these layers than the other two conditions, the control and the impoverished. Does that make sense? We all see that? Okay. So when we wanted to understand memory more, because it's a very complicated phenomenon, we have to start out with a paradigm that's extremely simple to understand so we can get to understand the biochemical changes that happen as a result of learning and memory. So the simplest form of learning is single or non-associative learning. Okay? Which, for an example of that is habituation, which is a decrease in response as a result of a repeated stimulus. And we study this in a very simple organism called the Aplysia, which is a lazy sea slug that looks disgusting about this big. Okay, you can actually find it in California. Okay? Now the sea slug, if you keep poking it, the first time you poke it, it will withdraw itself under its shell to protect itself because it thinks it's under attack. But if you're like, I'm just going to keep poking you, it knows, it learns that you're not threatening its survival, so it stops to withdraw. That's called habituation. That is a form of learning because it benefited from previous experience that you're indeed not a harmful stimulus, so it stops to respond. Can everyone see why that would be a form of learning? Okay, so we can use something simple like habituation in a Aplysia, which has just a few neurons and big ones, to understand what are the biochemical changes that happen to allow this Aplysia to remember that you're not a harmful or noxious stimulus. Okay, so you can have what's called short-term habituation, where you have, that's caused by synaptic changes between a sensory neuron that's feeling your poke and the motor neuron that's making it withdraw itself under its shell to protect itself. And how that's done is that there's less neurotransmitter, increasingly less and less neurotransmitter between the sensory and the motor neuron. Okay? Let me know if you're not seeing the link between that and what we're talking about memory and learning. And there is what's called long-term habituation, where actually you have the less neurotransmitter released in the synapse, you actually have less retraction, and you start to have fewer synapses between the sensory and the motor neurons. So these are more permanent changes. Thus it's called long-term habituation. So with short-term, you have less and less neurotransmitter being released between the sensory and the motor. With long-term, you have actually fewer synapses, or synapses that die away. Here's a picture, okay, of the siphon when you keep poking it and actually withdrawing itself under its scale. Okay, so let's look at another way of representing this information, the short versus the long term. A stimulus, which is the poking to the siphon, is going to, with water or poking it or whatever, it's going to make it retract itself. And this is because there's going to be less neurotransmitter between the sensory neuron that's feeding the water on the siphon or your poke and making the Aplysia protect itself and withdraw itself. Okay? Now, with time, as you keep doing that, there's actually going to be less neurotransmitter released on the motor neuron. With long-term habituation, what you see here is, you see how you have here 3 synapses, 1, 2, 3? What you have here is these ones go away. You have fewer synapses and you only have 1. Okay? That's long-term habituation. Okay, so that's the simplest form of learning and memory, where we can get to understand a little bit of what's going on biochemically, but we can also go a step higher and look at something a bit more complicated and look at associative learning. What are the biochemical changes? What happens when you associate to stimuli? For example, we talked last time about the eye blink reflex. Puff of air to the eyes, it makes you want to blink. Okay? But if you pair that puff of air with a tone, a sound of a tone, so every time you hear the tone, you get a puff of air, then the sound of the tone by itself can actually make the animal blink. Right? Okay? Or the sound of the tone, in other words, becomes what? The conditioned or unconditioned stimulus?
Conditioned stimulus.
The conditioned stimulus, precisely. Right? Now, the unconditioned stimulus, which is what here again? What's the unconditioned stimulus? Yes, in the back.
The puff of air.
Exactly. Okay, it's going to be represented, that information is going to be represented via the trigeminal pathway. The conditioned stimulus, which is this acoustic tone, is going to be represented, sure enough, by an auditory nucleus. So after conditioning, these two activities are going to be paired like this. You have, before the training, you have the air puff and conditioned stimulus going to the trigeminal nucleus. You have the cranial motor nucleus that's going to make your eyes, the reflex, leading to the eye blink, and also this coordination is done by the cerebellum. This is normal without the sound of the tone. If you keep having the sound of the tone also, then this information goes to the cerebellum. At the same time, the information about the air puff goes to the cerebellum. Then over time, with training, if you get rid of the air puff, the sound of the tone itself will restart that same circuit, leading to the eye blink. Does that make sense? So Donald Hebb came up with this, what seems simple, but it wasn't simple at the time, is that neurons that fire together wire together. Okay? So two neurons, if they're always activated together, that they become connected and they become stronger. So if one of them becomes activated and maybe the other one is weakly activated, it actually has a bigger effect on the weaker neuron. And sometimes how we test that is by applying a tetanus, which is basically a volley of action potentials to one of the presynaptic neurons, and see what happens to the postsynaptic neuron. Okay? And this is one way to test what's called long-term potentiation, which is what we think is a proxy for long-term memory. Okay? We think that this long-term potentiation, which happens at synapses that are formed in a stable and effective synapses, we think that this is the proxy for what we call long-term memory. So here's an example. If you apply a lot of action potentials, a lot of stimulation to a presynaptic neuron, and then you measure the effect on a postsynaptic neuron, what you see here is that you get potentiation, meaning that the firing rate of the postsynaptic neuron has increased. Can you see that? Not only is it increased right after, it remains potentiated, it remains increased, look, up to 120 minutes, or as far out as they measured. So it's as though that the postsynaptic neuron remembers this information for, here it shows up to 2 hours after, but it has been shown to be much longer than that. And that's why we use this LTP, long-term potentiation, as a mechanism or as a way to study, or we think it is a proxy for long-term memory. I know the link is a little bit challenging to see, okay, but do we all understand this? Okay. We also know that long-term potentiation, the slide that I showed you earlier, everyone understands what I mean when I say potentiated. The postsynaptic neuron remains potentiated. You see that up state in the previous, you see how it stays up like that? Even though here at 90 minutes or at 60 minutes, the presynaptic is not firing anymore. It stopped firing. It's not getting excitation anymore. But it still remains in this potentiated elevated state for hours. It remembers. Okay? So we know that we see long-term potentiation in the hippocampal formation, in the hippocampus, and also in an area called the dentate gyrus, which is a part of the hippocampus. And the receptors and the neurotransmitter that's most related and that causes long-term potentiations are glutamate and its receptors, the NMDA receptor, along with the AMPA receptors. So both NMDA and AMPA receptors are receptors for glutamate. At low levels of stimulation, glutamate is released from the CA1 synapse. CA1 is a subfield of the hippocampus, so you can put above that hippocampus to make it easier for yourself. And this release of glutamate from the CA1 subfield of the hippocampus, you only get the AMPA receptors activated. So at low levels of glutamate release, meaning when the presynaptic neuron releases a little bit of glutamate, you only get the AMPA receptors activated and not the NMDA receptors. The NMDA receptors at rest or at low activation cannot really be activated easily because they have a block inside the receptor. They have a magnesium block inside the receptor. You can't do anything. You cannot activate the NMDA receptor unless you eject, unless you kick the magnesium block. Because it's sitting right there in the receptor. You can't do anything. The glutamate can't get in unless you kick this magnesium block. So the NMDA receptors are not going to respond until enough depolarization or enough stimulation happens to eject this magnesium. How does this happen? Is when you activate many nearby AMPA receptors. So glutamate gets in the AMPA receptor, it depolarizes the membrane a little bit, it keeps getting through all the AMPA receptors, many of them, it depolarizes the membrane enough that the magnesium, which is positively charged, is now repelled from the membrane. Why do you think it would be repelled by the depolarization? This positively charged magnesium. Yes?
Because there's an influx of NA.
Exactly, precisely. Depolarization means that the inside of the cell becomes more positively charged because sodium is coming in, Na positive. And because it's becoming increasingly positive, the magnesium, which is also positive, is repelled. It's no longer attached, it's no longer interested because now it's positive and positive. Okay? So with enough depolarization, because or courtesy of the AMPA receptors, the NMDA receptors can now be activated because it's the block, the magnesium block is ejected, and you can now have the NMDA receptor be activated. Yes, Sophie.
To stop everything you ever learn, like going straight into long-term memory? Is that why this is here?
What do you mean?
Like, because it seems like it's sort of difficult to get the magnesium to kind of move out the way. Is that a mechanism to stop us, like, remembering everything we've ever heard?
Not really, because this is very rudimentary, very crude, like very simple, going to something as large as what you're talking about. We're talking about a lot more changes. But I'll tell you why this magnesium block is very important. Why is it the NMDA receptor, activation of the NMDA receptor is so important? Because only when the NMDA receptor is activated, magnesium is ejected, not only does the sodium come in, which is what we expect of depolarization, but another molecule comes in that makes lasting changes that are extremely crucial for what we call memory, and that is calcium also comes in. Okay? Calcium will only come in once the NMDA receptors have ejected that magnesium and are free to be depolarized by glutamate. And once the calcium comes in, it activates a lot of intracellular enzymes that lead to a lot of changes, actual structural changes. For example, it can make the conductance of channels, it can make them more effective. So they can conduct, they can allow more ions to get in. It can position the receptors in such a way so they become more effective at receiving these ions. It can actually insert more receptors in the postsynaptic membrane. It can send a retrograde message to the presynaptic to release more neurotransmitters. Can you see how all of these changes are more lasting and they're structural changes? Okay, so here's a schematic representation. Glutamate is released from the presynaptic neuron. It wants to activate the NMDA receptor, but unfortunately there's a magnesium block, so it cannot do that. But the AMPA receptor is free to be activated because there's no block. So sure enough, glutamate activates it, which leads to the sodium coming in. The sodium coming in depolarizes the postsynaptic neuron, which then, and only then, can eject this magnesium. Okay? And you see here how there's some AMPA receptors that are actually inside the cell. They're latent, they're not active. These ones can be inserted back here once the calcium comes in, once the NMDA receptor is activated. So here, because of the AMPA receptor, because of the depolarization, the magnesium is ejected, and only then does, in addition to sodium, calcium comes in. And once calcium comes in, very important second messengers become activated, like the CAMK2, the protein kinase C, all of those there in the middle. And the activations of these second messengers will then lead to some long-lasting structural changes that then allows this memory trace. Such as, we have here, do you notice that there's one more AMPA receptor being inserted? So the actual postsynaptic area looks different. Also, do you notice that there's more glutamate being released, more red dots? This is a result of this calcium coming in and some intracellular changes that happen in second messengers that then sent a retrograde message to the presynaptic neuron to release more glutamate. Does that make sense? Okay, so we talked about the retrograde transmitter. We talked about all of this. And any synapses that are not going to be used, so they're not going to go through this, what will happen to them is that they will fade away as they become weaker and they don't exist anymore. Okay? So synapses, that's a very dynamic process. Ones that are not used will just die away. If you don't use it, you lose it. Okay, so here's an example. Let's look at, on the left side, neurons 1, 2, and 3. What do you notice about the firing rate? Each stick, or each one, or whatever, like each line there represents an action potential. Okay, each line represents an action potential. And the spacing between them is kind of like the pattern over time. So what do you notice about 1, 2, and 3? Yes, Victor?
It's more harmonized and it has a rhythm to it.
Exactly. The three neurons are firing action potentials in synchrony, or as you said, in harmony. They're all doing it at the same time. They're synchronized. And what is the effect on the postsynaptic side? So on the left you have the presynaptic side. On the right you have the postsynaptic side. What is the effect of this firing and the synchrony on the postsynaptic side? What is it leading to in the postsynaptic side? Is it successful in turning on the postsynaptic side? You're nodding yes. Why do you think it's successful? How does it look successful?
Is it also because it's synchronized as well?
And also it's successful in producing action potentials in the postsynaptic neuron. There's lines, right? And not only that, but also the postsynaptic neurons have also moved collaterals, additional synapses. Do you see that in the postsynaptic side? Now let's look at the bottom two neurons on the presynaptic side. Okay? We see that the action potentials are not really in harmony. They're not firing that many action potentials. Not only that, even the action potentials they're firing, is it having an effect on the postsynaptic side? No. So because the synapse is not successful, what happens to them over time? They're going to be lost. Exactly. They're going to die, they're going to be lost. Does that make sense? Okay. Why am I saying that LTP is a proxy for long-term memory? Why am I saying that long-term potentiation is a proxy for long-term memory? Well, we think, at least from correlational study, that the time course of long-term memory is very similar to the time course of long-term memory. So here's one piece of evidence. Also, from somatic intervention experiments, which you should know what that means from chapter 1, is that pharmacological treatments that block, so anything that you inject the animal with that blocks long-term potentiation also impairs the animal's ability to learn. Okay, here's another piece of evidence. Also, we know that mice that overexpress, so where you genetically make this mouse overexpress NMDA receptors, they also tend to have enhanced memory in LTP. Okay, so they've actually made like mice that overlearn very fast and efficient. Okay. We also know from behavioral intervention experiments that training an animal in a memory task, that when we sacrifice the animal, or if we look, or if we're measuring LTP, that we can actually induce LTP by training the animal. So all of these together are convergence evidence that show that long-term memory might be a good, long-term potentiation is a good proxy for long-term memory. Okay, does that make sense? Yes, Sasha.
All those three were for long-term memory only?
Yes, yes. Okay, we're not talking about iconic memory. This is long-term memory. Okay, so does that, all of that make sense? We're going to move to kind of like a little bit of a different module. So does the memory stuff make sense? Okay, let's look at prenatal development, or how actually the nervous system develops. Okay, so the human embryo is going to develop from develops into three cell layers: the ecto, the endo, and the meso. Derm. Derm means skin. The ectoderm is the outer layer. Ecto means outer. Is going to actually thicken, and it's going to become the nervous system. So the outer layer of the embryo is what becomes the nervous system. And in the middle of that, it's actually a plate that folds upon itself, so we call it the neural tube. That forms also neural ridges that will become very important. The interior part of that folding over the neural tube becomes three big bulges: the forebrain, the midbrain, and the hindbrain. Okay? And an embryo, for the first 10 weeks, you call it an embryo. Beyond that, you call it the fetus. So here's a picture of the ectoderm, the endoderm, and the mesoderm. Meso means middle. You know that from previous things we talked about. So over time, you see that groove, that neural groove, the yellow with the lines, that folds upon itself, that becomes the neural tube. Okay, folds upon itself like that. And then over time, you see that the front of the neural tube differentiates into these bulges called the forebrain, midbrain, and hindbrain. And then each of these bulges further differentiates into other bulges. So for example, the forebrain is going to become the telencephalon, the end brain, the diencephalon, the middle brain, and then the hindbrain. Okay? So here's prenatally what the brain looks like. So do you notice that at the very beginning there is no gyration? So it's pretty smooth. Okay? But even at birth, even though the brain looks like an adult brain, it's not. Okay? And at birth, the brain, I guess infants look cute because their head is like one quarter of their body. And that's because at that time, humans, when they're born, we don't care about spending energy on the animal or the human to start running and walking. You all know that infants don't start walking as soon as they're born, while other animals do. And that's because most of the energy is dedicated to just the brain. Okay, and that's why the brain looks really big compared to the body. So brain development, you have different stages. First, obviously neurons have to be born through mitosis, cell division, which is what's called neuro for neuron, genesis, birth. And then once they're born, they have to migrate, they have to move to the final destination. Because they're all born around the serum area, the ventricular zone, and then they have to migrate out there to go to where they need to go in the brain. And then once they get to their final destination, they have to start looking like a neuron or a glia cell. So if they're a neuron, they have to start looking like a neuron by growing dendrites and axons. And once they do that, and they look like a neuron or a glia, they have to start making synapses, synaptogenesis. And neurons or an unmature brain, you have many, many neurons being born, more than you need. You have many synapses being formed, more than you need. And what ends up happening is that neurons that are not used die, or neurons that don't make synapses, they die, a healthy death. And after this massive death in pruning, what happens is the synapses rearrange themselves again. Okay? So the middle part is is the ventricular zone that I mentioned, where all the neurons are then going to migrate from there to where they need to be. And then once they get there, they start to look like neurons or glia, cell differentiation. And then once they have differentiated into neurons, they start to make synapses. And because we have too many cells, more than what we need, and we have all these inefficient and efficient synapses, well, the ones that are efficient and are used are going to survive. The ones that are not will die. And then you have rearrangement, a more mature rearrangement of synapses. Okay? So again, going over the stages again a little bit more slowly. Neurogenesis is the birth of neuron or production of neurons. That's going to happen through mitosis, the division of cells. And this is going to form in that ventricular zone, that yellow center, that was line that I showed you in the earlier slide. And then this can become a neuron or a glia. And then after that, now the entire neurogenesis state does not have to change before cell migration begins. So once the neuron is born, it can start migrating, for example. Okay, moving away from the ventricular zone or layer, going to where it needs to go. Now, how does it know whether it should be a neuron or a glia or where should it go? It's because it has its own program, genetic program, that makes it produce certain proteins that will make it grow dendrites or axons. Okay? And then it becomes differentiated, begins to make synapses. And the cell-cell interaction between a cell and another cell can actually affect each other's ability to differentiate. Okay? And when we say stem cell, really what we mean is undifferentiated cells, or a precursor cell that has not determined yet whether it's going to be a glia cell or a neuron cell. Or even a much more undifferentiated cell is a cell that hasn't even determined whether it's going to be a brain cell, a kidney cell, or a heart cell. But the only problem with planting stem cells that are completely undifferentiated like that is that you can potentially have a side effect of cancer. Okay? So it's much better to try to inject a stem cell that is a precursor neuronal cell. So that's destined to become a neuronal cell. Do we have adult neurogenesis? So once all of this is done, can we actually have new neurons being born? Because we lose neurons every day. And the answer to this question years ago was no. But now we know that there is new neurons in adults, but in few areas. So for example, in the dentate gyrus of the hippocampus, remember I mentioned that earlier, which is very important for learning and memory, you can actually see birth of new neurons there. On top of your nose, there's actually olfactory neurons dangling that are exposed to the environment, those ones die and new ones are replaced very often. But pretty much, other than that, if you lose neurons, they don't come back. So for example, if you do drugs like alcohol will kill neurons, those neurons don't come back. Okay? So really, other than that, this is really the exception. It's not the case that if you have neurons that are damaged, you can actually get them back. So here is basically a picture. The red labeling there, the BRDU, is a label that tells you what areas have DNA in the cells that are about to divide. So it tells you what cells are about to divide or about to be born. The green or the new N is a dye, the green dye, that tells you that this is a neuron. Okay? So the coexistence of BRDU and UN tells you the cells that are about to divide that are neurons. So basically birth of new cells. And what you see here is that this is a way to get to know if an area is able to grow new cells or not in adulthood. Now, I said earlier that you have many more cells than you actually need, and so you go through healthy cell death, and this healthy cell death is called apoptosis, which is very different than unhealthy cell death that, let's say, happens in Alzheimer's disease, which is called necrosis. So apoptosis is a healthy part of brain development that allows fine-tuning and that allows this change in synaptic arrangement of a more efficient circuit. Okay? And these cells come with their own genetic genes for death, for healthy death, if they're not being used. And similarly, neurons that are being used and are efficient release at the same time what's called neurotrophic factors that maintain their integrity, that help nourish them so they can live and survive. Does that make sense? So the cell comes with this stuff programmed in it. Genes for death if it's not efficient, neurotrophic factors to be released if it is efficient. Efficient means that it makes synapses with another neuron. It's able to make synapses if it's being used. How does it know if it's being used? Well, if it's releasing neurotransmitter, it's being used. So along with that, it releases neurotrophic factors that helps maintain the integrity of that neuron so it lives longer. Does that make sense? Okay, so here is in chicks. If we look at the number of motor neurons or cells, you see that a big drop, a big cell death at about 10 days. Okay? Same thing in humans, that you see this pretty severe drop at about 25 weeks or so. Massive cell death. And also around adolescence, you get the same pruning effect where a lot of cells die as well. Yes?
How are they able to count the number of cells?
They got some students who was really interested in going to grad school to do this. But typically what you do is you take an area and you generalize to other areas. You probably didn't count all of them. Okay. Okay. So here's a model of action for neurotrophic factors, a target cell. You have this other cell that's releasing neurotransmitter. You see on the second one, the one that's not releasing a lot of neurotransmitter withers away and dies. Or if it's not releasing any neurotransmitter at all, you see cell death, where they just disappear and they die. Similarly, if a synapse is not being used, as in the last panel there, it just withers away and dies. So literally, if it's not used, use it or lose it. After this period of apoptosis or cell death, neurons grow to be more mature and elongate, and they have more elaborate, and what I mean by more mature, I mean they have more dense dendrites, they grow synapses, new synapses, just the circuit becomes more mature. Okay, which creates a new synaptic arrangement. And again, this happens again, apoptosis during adolescence. And that's why adolescents tend to be a very unstable time. Okay, because there's actual instability going on in the brain. Okay? So you, as a parent, it's very difficult to put yourself in your adolescent's shoes because you have different brains. So here's at birth, 3 months, and at 2 years. Look at how, this is cortical, the different cortical layers, 1 to 6. You see the differences in the synaptic arrangement, the maturity of that synaptic arrangement, and the complexity of that synaptic arrangement. Okay, the number of synapses in the auditory, visual, and prefrontal cortex from birth to age 10 to adulthood. Do you see the number of synapses? You see a peak at about 1 year old. Actually, there's 3 different peaks depending if you're looking at the auditory, the visual, and the prefrontal. But do you see how the visual one, the red one, peaks a little bit earlier? And then the auditory one has a more flatter peak. But what do you see the prefrontal cortex? What do you see, prefrontal cortex? Is the peak earlier or later?
Later on.
Later on. In fact, the prefrontal cortex is not really completed until, well, some people say 18, some people say early 20s. I like to say early 20s. And remember, we said before, the prefrontal cortex is important for what kind of things, what kind of behaviors?
Decision making.
Decision making, impulsiveness or lack of impulsiveness. And that's people who have ADHD have what's called hypo, meaning little or under, frontality, meaning under activation of the frontal cortex. So the prefrontal cortex is really not fully developed until, I would say, early 20s. So really before that age, people should not be making massive, serious decisions that will affect the rest of their life. Okay, and then what do you see here at adolescence? The number of synapses. Do they drop? Do they go up? Are they the same? Exactly, they're decreased. Okay. Okay. Every cell comes with its own genetic information. Okay. Now I have a question for you. So every single cell in your body, whether it's in the kidney or the brain or the heart, has the same identical genetic or the same number of chromosomes, right? The same number of chromosomes. How does a cell know to become a liver cell or a brain cell or a heart cell, even though they have the same number of chromosomes? Yes, Sophie.
Isn't it based on what the cells next to it are doing when it develops?
Okay, what else? Let's think more along genes. So they all have identical chromosomes. How does a cell know to become a liver, kidney, heart, brain cell? Depending on which genes are activated. Okay? So a brain cell, the genes that are pertinent to a brain cell are going to be activated. But the ones that are pertinent to a heart cell will not be activated. Genotype, to differentiate it from phenotype, is the sum of all the intrinsic genetic information, so the actual nucleic acid sequence. A phenotype is actually how this genetic information, how you're actually seeing it physically. So you might have the genotype for a certain disease, but you might not, phenotypically, you might not be expressing it. Does that make sense? So a genotype is constant, and that's what gets inherited generation after generation, but the phenotype can actually change. Okay? So a phenotype is definitely going to take the genetic information into account, but it's also going to be responsive to extrinsic factors like the environment and other things going on. Okay. So for example, you might have a genetic disorder where you have the genotype for phenylketonuria, which is a disorder that makes you not metabolize a certain amino acid that is prevalent in many things. and if you happen to have this amino acid, the phenylalanine, and I don't know if you've noticed some grocery stores where right outside products contain phenylalanine. I don't know. Next time, I'm sure you will notice this. And because people who suffer from PKU, if they ingest or for some reason have something that has phenylalanine, which is not uncommon, they can actually develop severe intellectual disability, where they become severely mentally disabled. But this can be rescued if you just provide a diet that does not contain phenylalanine. Okay? So here the person or the child has the genetic equipment to become mentally delayed and severely, actually, intellectually disabled. But as a result of certain changes in the environment, phenotypically, the child might not actually express this because you have made changes in the diet. Is the difference clear between them, genotype and phenotype? Okay, epigenetics is, did I show this graph here or I didn't? I can't remember. Okay, I'll think about it. Epigenetics is a study of how gene expression actually interacts with the environment. Okay? So, for example, maternal care can actually be a very important epigenetic factor. Did we talk about psychological dwarfism here? We did, right?
Yeah.
Yes, where I showed that graph and I showed the height, the psychological dwarfism, and I showed how it can be reversed by just having these children being adopted in other families and how they catch up to their peers. That's an epigenetic factor. Okay? So having mice that are genetically identical, but have them raised by different mothers, will they actually show differences in behaviors, even though they share the same genes? How does that happen? How does actually epigenetic work? Well, there's what's called methylation, which is an actual chemical modification of DNA that does not actually affect the nucleic acid. So the actual sequence, the actual letters, the AT, that actual nucleic sequence does not change of the DNA, but makes the gene less likely to be expressed. So let's say you have a gene for psychopathy. Typically, it's a collection of genes, it's not just one gene, but let's remain simple. And we definitely know that there is genetic contributions to psychopathy. And so you have this genetic background or tools that destine you to become a psychopath. But what happens is that we provide a certain environment that is nurturing, a certain environment that's suitable for someone like that. Then what happens is that you might actually get the DNA to be methylated where you're less likely to express these psychopathic genes. Does that make sense? Okay. Obviously, the opposite can happen as well. So, for example, poor maternal care can actually induce methylation of glucocorticoid receptor gene in the brain, which is a stress hormone, cortisol. Which is going to make the animal more responsive to stress, meaning that because of this poor maternal care, which led to very real changes because you have methylation of the DNA, the animal becomes more responsive to a stressor. So while maybe other animals that did not have this poor maternal care find the stressor benign, psychologically benign and not stressful, this animal, because of its background on the poor maternal care and the methylation, finds the stressor very stressful. Does that make sense? Okay, this is like extremely serious. Okay? Because that kind of says that there's an onus in society and responsibility for like changing the actual chemistry of this organism in front of them. And these changes are lasting changes. So you can't really catch up with this child in adolescence or as an adult and say, okay, I'm here for you now. Tough luck, too late. Okay? Same thing when I talk about these animal studies is because we can't really do this invasive things in humans, but same thing happens in humans. Okay? The same gene that's important for this cortisol, in humans it's called cortisol, not glucocorticoids, become also methylated in children who suffer from childhood abuse and actually make them commit suicide. More likely to commit suicide. And it's also, this methylation is also prevalent in people who suffer this and who are suicide victims because of child abuse. Okay, I'm hoping to really get you to appreciate the seriousness of this. Do you have a question? Okay, does this make sense? And the earlier the abuse, or the earlier the trauma, whatever the trauma is, I mean, here we're mentioning child abuse, but a lot of other horrific things can happen to children. The earlier the trauma that happens in childhood, the more severe the effect is and lasting that effect is. While later traumas, you might be more resilient to it. Now, this methylation is going to be inherited by the next generation. So the key things here, we're talking about epigenetics. There's no changes in the actual DNA, in the actual sequence of the nucleic acid that makes up the DNA. There's no actual changes there. But this appendment of a methyl group can actually change how this gene is going to be expressed or not expressed. That will lead to very serious changes in behavior. Does that make sense? Okay. All right. We know that if somebody's showing memory impairments, that if we look at the brain, sure enough, there's shrinkage, particularly in an area that's important for learning and memory, i.e., what is this area called? The hippocampus, exactly. Okay? Now, the shrinkage of the hippocampus happens normally with aging. So as we age, the brain shrinks. We lose brain cells. That's normal. But if this shrinkage is a lot, and if we lose a lot of neurons, that's not normal. That's called Alzheimer's disease. Okay? Also, we know that the hippocampal formation volume is associated positively or correlates positively with memory, with certain types of memories. Okay? So here is, you see the hippocampus here, but that's where the hippocampus is. Dementia, which, I mean, dementia, when you say dementia, people think of Alzheimer's disease, but there are many different types of dementia, and the dementia exists in other types of diseases as well, not just in Alzheimer's disease, is a drastic failure in cognitive ability. The one that's more noticeable, or most noticeable, is the memory failure and disorientation. Alzheimer's disease is a serious form of dementia, where you have memory loss of recent events. You start by losing the present, a little bit of the early, close past, and then as the disease becomes worse and worse, you start to lose more and more memories in the past. But I don't know if you have a grandparent who suffers from Alzheimer's disease, or if you volunteer somewhere and you would sit with them and they'd start acting like it's still World War I, and they would start recounting stories. Like they're very bad, like they've lost all the recent memories and all they have is the very, very distant memories. Okay? And we talked about that before, that very distant memories are the ones that if damage were to happen, they tend to be the ones that are most protected. That you typically will lose the recent, the present, and the recent memories. You start with those. Okay? And then eventually as the disease becomes more advanced, it becomes very impossible to have a conversation with them because they're severely disoriented. They're just not in the context. It's very difficult to have a conversation with them. What happens cellularly that causes these changes? We know that there is a couple of genes that are important in Alzheimer's disease that code for obviously proteins. And in most neurodegenerative diseases like Huntington's disease disease and other diseases, there's typically a misfolded protein. So one of the crucial things about a protein is how it folds. If it misfolds, it tends to aggregate. It tends to aggregate and clump together, which causes severe pathologies such as Alzheimer's disease. So an example of this protein is amyloid plaques. This beta amyloid starts to misfold because instead of being cleaved in a certain size of amino acids, it gets cleaved too long. It has a couple more amino acids, so it clumps up. Same thing happens to another protein called the tau protein, which creates neurofibrillary tangles. It creates tangles between neurons. While the beta amyloids build up, it tends to build inside the soma or inside the cell. Okay? These built up or these clogs tend to aggregate more so in the frontal, the hippocampus, thus the learning and memory problems, and some limbic areas. Also, the acetylcholine or cholinergic neurons in the basal forebrain start to die out. Thus, in the past, we, in order to, or in the hopes of treating Alzheimer's disease, we try to provide acetylcholine because those neurons start dying. Unfortunately, though, the improvement doesn't last, maximum maybe 6 months. Now, how do you for sure make a diagnosis that this person has Alzheimer's disease with definitive 100% is when the person dies and you analyze their brain. And then you look and you do a PET scan and you see indeed these beta amyloid and these tangles in the brain. Although now you can actually do a PET scan when the person is alive by injecting a radioactive tracer to look at a picture of the brain and the distribution of these clogs, these amyloid clogs and tangles. Yes, Jane.
I kind of forgot, so how do the folds happen where? And then the buildup happens because of
Okay, so a protein has amino acids, right? There are amino acids holding hands, and then it folds in a particular way. The fold of the protein gives it its shape. What happens sometimes that causes pathologies, such as the one in Alzheimer's disease, is these proteins misfold. This misfolding is going to give them the wrong shape. As a result of this misfolding, they start to aggregate. They start to aggregate in places, which prevents healthy synapses and healthy information processing, okay, which might lead to Alzheimer's disease and dementia. Two of the proteins that misfold we think is very important in Alzheimer's disease is the amyloid beta proteins and the tau protein. The tau protein misfolding causes these fibrillary tangles. The amyloid buildup causes these plaques to form. Where do these things aggregate most? They tend to aggregate most, meaning where do they cause the most pathologies, in other words. They tend to aggregate in the frontal area, limbic system, the temporal, but also in the hippocampus, which we know is very important for learning and memory. Okay, does that make sense, Jane?
Yes.
Okay. Here's the picture. The brown are the plaques that are caused by the aggregation of the beta amyloid protein. The tangles are the yellow, which is caused by the tau protein. How I used to remember this is tau begins with T, and tangles begin with T. But anyways, and then the other one is the other one. Here's the technique I told you about, where you inject a radioactive material to track the beta amyloid. You see the hotter colors, the red, yellow, orange, that means high levels. The cooler colors are the blue. What do you notice between controlled subjects and Alzheimer's disease? Is it more hot or cool colors? Hmm? Exactly, high what? Well, this one happens to for the beta amyloid. So you see a lot more beta amyloid in somebody who suffers from Alzheimer's disease. And there's another group that's in the middle that has not really reached Alzheimer's disease yet, but they have cognitive impairment. We call that group typically in research mild cognitive impairment. And we call them MIC patients. Sorry, MCI patients. Why did I say MIC? Okay, never mind. Yes, mild cognitive impairment. Okay? But when you do a normal person, you see mostly, or actually you don't see really any of the hot colors. Okay? Now, this is invasive, so it's like, oh, I'm just going to go and get it done. Well, you are injecting radioactive tracers, but when you are somebody who suffers from Alzheimer's disease, this is, this is a low cost to pay. Does that make sense? Any questions?